Alright, so what we want to do, we want to look at the basic ground plan of insects in this lecture. So this one gets you to the outside of the insect and some of the sort of areas you'll be looking at in insects. And your handout has nearly all this information here for this lecture, plus some of the slides. So, skeletal ligament, the outside. So, things like antennas as well, out there. That we want to take a look at. The first thing I want to do then is to look at the basic ground plan of an insect. I'm just going to see where my notes are. That's all good. Okay, let's take a look at the basic ground plan of this. Original insect would have had 20 body segments back to those 300 million years ago. Okay, but what's happened is 6 of those segments have fused themselves to the part of the head. Okay, and then we've got 3 segments here, each with a pair of legs, which we call the pro, the meso, and the metathorax, which is, you know, the front, the middle, and the back thoracic segment, with both the meso and the metathorax having wings associated with them. So there's 3 segments there, and then there's 11 segments in the abdomen, so 11 plus 3 plus 6 becomes 20. Now you do get some level of fusion in the back few segments here when we're looking at insects, particularly around forming some sort of the reproductive things like ovipositors for laying eggs and things. That might mean that the back 2 or 3 segments may well be fused. But that's the basic ground plan that they started with, but the insects have undergone a number of different fusions in order to achieve what we do see today with a head, a thorax, and an abdomen. And again, if we want to have a look outside of the textbooks, we can have a look at Jimmy the Cricket as our first non-insect example, if you like. Poor old Jimmy's only got 4 legs. I don't think he's got an abdomen at all. He's probably only got 2 segments of his thorax and a head, so he's about 8 segments only. So not that successful. Certainly wouldn't be able to breed. B-movie, which was out more recently, is Jerry Seinfeld. Again, he's looking a bit dubious. Again, 4 pair of legs. Got antennas up this time. Gym boots, of course. But probably looking at some reduction there in thorax and the abdomen. We want to go to better. This is it. This is the Ant Bully movie. I think it's Ant Bully. And the reason that this is better is it turns out Tom Hanks did this movie, and when I was in California, the officers there were with a guy called Larry Hanks, who turns out to be an entomologist, in fact Tom's brother. So I reckon Larry would have had a little bit of input saying to Tom, you can't make stupid insects. So you can see here the representation of ants is pretty good in Ant Bully, probably the only example where the insects haven't taken so much artistic license, thanks to Tom's brother, I suspect. I don't know for sure. Okay, let's now have a look at the exoskeleton, the hard outside part of the insect. As I said already in the first lecture, it's really important because it stops desiccation. It's a rigid outer shell, it's a support for muscles, covers all those internal organs, but it's not always just like a nice smooth surface. It can have wrinkles on it, it can have hairs on it, it can have little sort of invaginations where muscles attached to on it. So it's not necessarily, or folds if you like, invaginations or folds, it's not just necessarily a flat surface. However, it is made up of a number of different parts, and I want to go through those parts. In general, though, if we took, I'll start going through the parts and then go the other way. So let's start from the inside. So here's the blood and guts of the insect on this side here, and the first point we have a basement membrane here, which allows some exchange between the hemolymph or the blood and these cells here of the external skeleton. So that's just a barrier, but there is some exchange. The next one here is a single cell layer called the epidermis. Okay. Now the epidermis is got a couple of roles. One I'll talk about when we get to the process of growth and molting, but this one I'll immediately mention with this figure is that it has glands in it that produce both waxes and cements that go up through there and into what we call the epicuticle here. And I'll talk about the functions of those waxes and cements. So it provides wax and cement up to the epicuticle. Then we have a region here that's called the cuticle, okay, which is the endocuticle, the exocuticle, and the epicuticle, 3 parts. And the picture of this is on your handout there. So those 3 parts. The cuticle itself is a large component of an insect's body weight. If you were to dry an insect, 25% of its body weight would be made up of cuticles. So it's a very large component, like skin is a large component of ourselves. But it lines not just the exoskeleton, it does line other regions in the body. It also lines parts of the fore and the hindgut. Cuticle produces the wings and also produces the tubes for the respiratory system. So it's quite important in quite a few areas. Now within those cuticles we have 3 types, as I said, endo, epi and exocuticle. I want to go through each of those independently and talk about them one by one. So first one is the endocuticle. Okay, this endocuticle layer here is a soft layer. Okay, so it's flexible. Now if we have a caterpillar, for instance, you realise the outside of a caterpillar is very flexible. Okay, so it's largely made up of endocuticle with only a little bit of exocuticle present in it. Okay. So it depends on the region that it's deposited, but endocuticle enables that flexibility, okay? It's also very important when we come to molting, but I'll talk about that when we do come to molting. But it's not sclerotized, it's not hardened. Then we get to this next layer here, the exocuticle, okay? Now the exocuticle is tanned chitin, okay? So it has been sclerotized through the product of a reaction of chitin with quinones. So it's very hard and it's very resilient. So it's that hard, tough, flexible outer shell that we think of with insects, but it's particularly strong around the head, protection of the head, and particularly strong in mouth parts of some larvae. For instance, termites in their mouth parts have minerals associated with them, including something like, I think some of the termite species have about 20% zinc into there, which enables them to be really, really strong, and that's why they can get through wood and other things that you don't want them to get through. So very tough, very strong, that exocuticle can be. Okay, if you took an insect and you boiled it in, it died, let's say an insect died, and you boiled it in potassium hydroxide, all the soft tissues are dissolved, and you just see the exocuticle or the sclerotized parts of it when you look at it. Why would you do that? Well, just to prove, firstly, that how much exocuticle there is and how much is endocuticle, but the other thing is that it's very useful for looking for characteristics, and we don't have to do this in our labs, of flies, to identify flies. The genitalia has cuticle on it, and flies, and the male genitalia in flies can be very important to identifying species in flies, unfortunately, because flies are a bit of a nightmare in general. Okay, this layer on the outside then, this is the outside here, is the epicuticle, right? Epi for outer. And this has both a cement layer and a wax layer on it. Remember, these are fed by the epidermis, bringing these up. Now, the cement layer obviously helps provide some toughness to the outside, and the wax layer makes it resistant to water and other solvents. So in very dry conditions, they can alter the level of wax. So you've got more wax or less wax there as you might need it. So they have the ability to vary that. And the wax also reflects solar and ultraviolet radiation. The other thing that we generally find in the waxes, and it's being more and more discovered when people are doing research now, is that it also is where a lot of chemicals are embedded. And these chemicals are known as cuticular hydrocarbons, and they are very important in insects identifying other insect species when they are in physical contact with each other. So particularly relevant in some social insects. If you like, it's the armpit of the insect, the identification, species-specific identification contained within those particular hydrocarbons. Okay, so that is the epicuticle. Okay, now let's think about this unit. Oh, what's my next slide? Let's have a look at that. Okay. The outside of it also has potential to have colour embedded in it. There are two ways we can have colour in insects. There's a structural colour and a pigment colour. For the structural colour, it's the arrangements of small microplates, if you like, on the outside of the insect. Generally these become metallic when you look at the insect. You see metallic reflective colours. So here's a wing of a morpho butterfly, and when you look at it under an electron microscope you can see all the orientation of these, and this colour comes through the scattering, interference or diffraction of light from the surface of these different plates. Okay, so that stays with that insect for the whole time, as opposed to pigment colour. Now pigment colour is the result of certain chemicals. Melanin is one of those that can be there, that you might be familiar with, that are present as granules in that epidermal layer, in that waxed layer, and that provides the colour. So aphids, for instance, you get green aphids, red aphids, other coloured aphids, and that's partly the diet they're on. If they're on rose bushes, they tend to be more red. If they're on something else, they tend to be more green. That's a pigment colour. And you'll notice when you're collecting your insects and you kill them, that you'll quite often see after death that the colour goes, and that would be because it's pigment colour, not structural colour. Whereas your butterfly wing stays colourful the whole time. It's not a pigment colour, it's a structural colour. Okay, so you've got this hardened outside thing's really good, but the complication you have with that is you've got to grow. Okay, so how are you going to get bigger and have this outside skeleton still exist in order to achieve that? Generally, insects go through a number of instars in the immature stages, or stages. So 3 instars up to about 11 instars, depending. We'll talk about that in other lectures. So you've got at least 3 molts that you've got to get through to get to be an adult, depending on the species. How are you going to achieve that? Okay, so this is where those epidermal single layer cells become important. Okay, so these are the site where they produce what's known as a molting fluid. Okay, so the epidermal cells start producing a molting fluid, and that molting fluid starts to seep into the space above the epidermal cells here, into the endocuticle. So here we can see that it puts down a new epicuticle because it's divided to achieve that. There, a new epicuticle, and then the molting fluid is sitting above that. Then, through a process of enzyme activation, the molting fluid becomes active. And once that's active, it starts to eat away all the soft endocuticle. Okay, that disappears. So your endocuticle is disappearing, and you see it digesting, and there's the old membrane there as it molts through. It gets to the point where only the old exocuticle, that hardened bit, is left on the outside. Okay, and here you're getting a new new endocuticle developing. At the moment it's called prior to cuticle, but it is the new developing endocuticle. So moving through there to that point, and then we have this gap here, if you like, where the fluid's redigested, which is like an air gap between there and the old exocuticle. So how do you get out of that? Generally, most insects will have a what do you call it? A weakness in the exoskeleton, a point line where they'll actually come through. So in the case of this cicada here, coming out of its last nymphal stage, is down here through the thorax, and it will come through there. How do they actually start to become bigger? The most common approach is they start gulping air, okay, and they start expanding out and out and out. So you sort of, when you're a nice loose sack, you try and get bigger and bigger and bigger as you can, okay, to get as big as you can before you start to become hard. In that process of coming out, the other way that you'll notice, because they're very soft, they couldn't walk or do anything at that stage, you'll notice they often use gravity. I've got this one upside down really, but they'll often use gravity to help them come out. They're just hanging from there for a period of time until they start to harden. So they have a very vulnerable window every time they molt. So 24, 48 hours before they become hardened and they get colour back up. So that, you don't see molting insects that often because they go off and hide somewhere to do that, because, you know, for 24, 48 hours you're going to be vulnerable to being attacked. Okay, the other thing that I do get quite a few phone calls about is albino insects, and 100 times out of 100 albino insects are just newly molted insects. There's nothing particularly profound about an albino insect. It's just that someone has come across one that has just newly molted. So that's your process of getting out from the old exoskeleton to a new exoskeleton. Okay, let's take a look at the head. Okay, we have 6 areas of the head that we said have existed. I'm going to find what I said on your notes here compared to what I'm about to say. Where have I got that? Okay. I don't have that there to actually mention. Alright, so we've got a first segment would have been made up of this part here, which is called the labrum. Now, I like to remember that as R for roof. This is the top of the mouth parts as you look down. So the roof of the mouth parts, so things don't go flying up. Then the next segment would include the antenna of the fused segments, okay? And the next segment then after that would be fused, would include the compound eye and the majority of the head. And then associated with these mouth parts, with those fusions, we have certain mouth parts. So we've got the first and the second and the third, and then the fourth fusion has something called the mandibles on it. So here you can see a mandible, and we'll see this in different angles in a moment. But the mandible is the chewing part. It's like if you think of the knife and fork set, it's the knives, okay? So it's the cutting, chewing part. So anything that's got to do a lot of chewing will be having quite prominent mandibles. The one I mentioned, the termites, that is reinforced. Then we have the next segment here, the fifth segment, with what's known as the maxilla. If they are the knives and the maxilla are the forks, it's the handling, like a fork is, of the food, alright? But it also is loaded, as you can see these pellets here, with a lot of sensors on it, so that they can actually taste their food properly before they actually ingest it into the mouth. So a lot of chemical sensors on there for tasting the food. And then the last part, which is associated with the sixth fused segment, is the labium, or the lower part, okay, of the mouth. So we can get those 4 regions and the other fusions to get the head. With the mouth parts, we can look at the way the mouth parts are oriented to try and get some idea, without knowing much about insects at all, about what their diet might be. So we have 3 different orientations of the mouth parts that are possible. They have a funny name, but they're orientations that I think are fairly self-explanatory. So the first one here on the left, the mouth parts are facing downwards. Okay. So if you're a grasshopper, let's say, sitting on a leaf, that's where you want your mouth parts, down there for just feeding on that leaf surface. So we'd expect a lot of these to be either omnivores or herbivores. Okay, if we've got our mouth parts facing forward, what sort of lifestyle do you think that would indicate? Burrowing? Sorry? Yeah? Sorry? What if you had your mouth parts out here? What would you be doing with them out front? Yeah, catching stuff. Predatory, okay? So if you're seeing a museum specimen, you don't know anything about it, and they've got the mouth parts oriented out here, likely to be predatory, okay? Okay? The last one, so that's called prognathous, or forward-facing, and this is a mouthful, isn't it? Opisthodorhynchus. But it is a long mouth part that goes backwards, okay? So it's orienting backwards. These are generally only in insects that have a long sucking mouth part. Okay, so there's three different orientations to give us a bit of a clue about what that insect might actually be for diet without knowing anything else. We want to have a look at that from a few angles and different points of view. Here's the prognathus, the predatory ant. Look at those mouth parts sitting out front. You know, that's great for running around and trying to capture your prey. So if you're predatory and you're an ant, that fits. If you want to think of an analogy, try and make you remember this, think of this gentleman here. Prognathus, forward facing, right? Obviously predatory, possibly with a female equivalent be Angelina Jolie there. Okay, so forward facing, predatory, beware. Okay, let's have a look at those mouth parts now, when we sort of blow them out and just have a look at them as the most simplified form. So this is the ground plan mouth part, if you like, series of mouth parts. So we have, here's your head, okay, compound eyes, etc. Here is your labrum, your root, okay. Let's see, you can see it here on this pretty wonderful looking fetal head. So your labrum or your root. Here are your mandibles. I said you're cutting with a cutting edge on them. There are your mandibles there. Okay, you can see those. And you can see ectognathus. Remember I said exposed mouth parts. Look at that. Look underneath, you can see them all. Count them up. Okay. Here is your maxilla. Okay. That's your knife, that's your fork set. Okay, with maxillary pouch for tasting. Okay. So there is quite a lot of tasting done by that, but there is also tasting done in other ways. Maybe I'll reserve that till I talk to that specifically and why that might be important. If you go back to why would I need to learn about molting of insects when I did molting? Well, the most target specific and some of the best insecticides have been developed by mimicking molting hormones, okay? So therefore, if you can mimic that hormone, the insect can't properly molt, and then they have a control of it straight away, okay? So understanding that process has led to the development of new ways of attacking insects. In this case here, I'll give you an elaboration about tasting in a moment. Okay, the last bit here we have is the labium, which is here. The labial palps are here, so there's some tasting. But you often also have another little part here, depending on the insect, which is called the hypopharynx. So this is in the generalised ground plan. We'll see this hypopharynx sort of around here somewhere. And that is the equivalent of like the tongue, if you like, in a way. So we've got labrum, mandibles, maxilla, labium, and hypopharynx. Okay. Now let's have a look how insects have modified those. And this gives you great clues about what that insect might be doing once we know what the modifications are. Okay, this is the first option. Modify your mouth parts. Look how weird they've gone now. Well, piercing and sucking. This is the Neptera, the true bug. Piercing, sucking, or a group. Okay. So your mouth part like that, we'll be able to understand a lot about it. Sometimes they're mostly herbivores, but they can be predatory too. Let's have a look how they've gone about getting piercing, sucking mouth parts. Okay. What the typical bug, in this case a cicada, has done is it has made its mandible into an elongated stylet, and it's also made its maxilla into an elongated stylet, okay, so like a needle. And then its labium is like a sheath that goes around that and keeps it together inside there. We have quite a lot of musculature in the head because it's going to need a lot of muscle for sucking up from there, and also a salivary duct you can see in here. So if you look at it side on, there's your mandible and maxilla, and there's your labium sheath there. So when we put that as a cross section, you can see they interlock all together quite well, and you have both a food channel and a salivary channel. Okay. Now the food channel is one thing. The salivary channel can be a real interesting box of tricks in terms of how they dissolve food for uptake, but it also can be the point where diseases that might get into, viruses and things that might get into insects, then transmit themselves down through the saliva into a plant or into a host, okay? Malaria being a prime example in a medical case. That means it becomes a vector of diseases. So it's that salivary gland, that salivary channel that becomes important. So they're using this mandible and maxilla sort of in a laid like this, burrowing their way in. But we'll talk about it in more detail in some other lectures. Piercing sucking, we've got a few more options too. This one here is mosquitoes. So you don't want to hit Botox because it's no good if you're a mosquito, but assuming you've got the correct human to suck blood on, this one here is the dengue fever transmitter, and some sort of malaria one. But you can see here again this sheet going down here. So these are going to have to go down a lot more into the host to drill down to get to the right capillaries to start sucking blood. So they are stealth, if you like, in how they actually get it. You don't generally notice a mosquito is biting you until it already is in the process of biting you. In that process of biting, what are they using for mouth parts? Okay, they've got even more. They've got more stylets, more pins and needles, if you like. So not only have they a mandible and a maxilla, which in this case is called the lacinia, they have a hypopharynx, that tongue, made into a long needle-like thing, and the labrum, also made into a long needle-like thing, all again sheathed in the labium. So 6 rods in there. And if you look how all these interconnect with each other, you still come up with a fairly large food channel, but you still have that little salivary channel there, which is important for piercing and sucking and getting fluids up in the system. So they do that. In their salivary, in their starting to bite you, they're going to put down an anticoagulant, all right, which makes that itch that you get when a mosquito actually has finished biting with you, to make that blood nice and flow up for them. So that's what the salivary channel is used for. Okay, change mouth parts again. Moths and butterflies. They have changed it to a siphoning mouth part. Now, not all moths and butterflies actually even feed. Some of them don't even feed as an adult. They don't even have functional mouth parts. But if they do, this is what their mouth part's going to look like. Okay. So this diet, again, is obviously a liquid diet. Okay. So you're never going to get, well, there is one exception, I should say. There is a vampire moth that does actually take blood. So, but on the whole, except for a vampire moth, if you're a tapir in Borneo, you'll be all right. This is just going to be busy taking nectar, if anything at all. Okay, so what's this siphoning mouth part? Fantastic looking tube. Okay, what is it made of? It's the maxilla. The maxilla has been totally modified into a coiled proboscis for sucking fluids. Okay, now if you want to look at the greatest example, I think, of this is with hawk moths. Now these actually hover, they're like the ecological equivalent of a hummingbird. They hover, they don't land on the flower, they hover, put their mouth parts in and take the nectar. So you don't see them generally because they're dust flying or night flying. But you can see here, this one here has a proboscis about 2.5 times its body length. So hover a fair way out, so we'll be getting into some pretty interesting flowers with a proboscis that long. Okay, the flies. You want to talk about with the flies? Yep, that's good. Okay, the flies. They've done something different again. Here's a fly. It's sitting there sponging up a sugar solution there. Now, as I said, flies on this mouth part here, which is known as a labellum, the big sponge here, there are lots and lots of sensory hairs, okay, on those, which taste the food that they're going to take up. So they're sort of acting like a big sponge, sponging up anything that they can pick up. So they're going and residing on feces, and they're having a bit of a feed on that, and then they come onto your salad. That's where you're going to get mechanical transmission of disease vectors through flies, because they're just basically putting a big sponge on there and sponging up whatever they can, and that dirty sponge goes on somewhere else. So that's why they can be very important in disease and veterinary problems. However, it's not just this mouth part that they taste with. Flies actually have sensors in their feet, okay? So when they're doing all these little things with their legs and everything else, they're cleaning up their feet, ready to lay down on something and have a bit of a taste. So they're tasting around with their feet, and then if they're going, oh, that tastes pretty good, I'll put my mouth part down and then sponge it up, okay? How does that become important? Well, If we're trying to control, say, fruit fly, we need to provide it with a bait that it will actually take up, that will take up a level of insecticide that it will actually eventually kill it. We've got a knowledge, and we can do this by tricking flies if you like. You can give them a solution, and you can see when it's just their feet in whether they extend their mouth part down or not. Then they're saying, yep, I like that. So if you're trying to mask a chemical in a food solution, then you can work out whether the flies will take it or not by placing them on it and seeing if they're going to put their mouth part down or not, and get that sort of bioassay, if you like, in order to be able to deliver something that they're going to pick up as a food for fly control. Okay, so flies, mouth parts, that labellum, and so you can see the maxillary palps, and the labrum is still sort of there, the epipharynx, but the labellum is the big change, the big shift in flies. We do have another option with flies though, and one of those flies that you might be familiar with is like horse flies, tabanids, march flies is another one. It's called cutting sponging, okay? So they just haven't got a labellum, they've actually gone and got the mandibles and the maxilla in particular back to being cutting or slicing instruments. Okay. So what a horsefly or a march fly does, when it's actually on you, you can see here the cutting instrument here in the sponge here, is it lacerates your skin till you can get blood starting to well out of that skin, and then it will sponge it up. Okay, so it's chopping it up and then sponging, chopping it up and sponging. Which is why if you've ever been in the snowy mountains or up in the alpine areas here, or Parts Mountain or something like that, and it's the march fly or the horsefly season, they're bloody annoying, and they spend a lot of time trying to bite you, but every time they start to bite you, you can feel them straight away. So, you know, you get them a lot easier than you do mosquitoes. So, although they're big, they're sort of slow and stupid in their approach for feeding. It causes a problem with feeding of livestock, of course, because they can't sort of move around and swat it. So if you're getting a lot of these feeding on livestock, then it can stress your livestock because of that nasty way of slashing up before they feed. Okay, if we want to go to the best examples of what mouth part you'd want if you really had a choice, it'd be those that are particularly the wasps have, the Hymenoptera, the bees, the wasps and the ants. It's a chewing, lapping mouth part. It's the best of both worlds, all right? So what they have done is they have still got their mandible here. They've got the labrum, everyone's still got their labrum, but they've got a mandible, so they can chew up things. So if you're a wasp like this one, or a worker honeybee, you can go mash up pollen or mash up prey with your mandibles, but then you have changed some of the other parts of here. You've got your labial palps here to a tongue, right? So there's a modification here to the tongue, which is sort of made up of different areas, and that enables you to take up liquids. So you can take either liquids or hard diets through that sort of approach to feeding. And I think I've got one to show you where they all come from, which is the next one. If you went through all these different things, and you start with the basic labium, so there's the labium's been modified here to the bee mouth part, the different mouth parts. So that's the basic, and then where it moves to all those different, some of those, not all of them, but some of those different designs. Okay, so mouth parts give you a pretty good idea of what their diet might be straight away, what they can and can't do. Really good. Let's have a look at a few other features just on the outside of the insect, which we will revisit in different ways in further labs. So the first one is the eyes. What do they see? We'll talk about that in another lecture in detail, but the really obvious thing in insects is this compound eyes on the outside. You can see them there, very spectacular looking things. They're made up of a number of different little cells called ommatidia. We'll talk about that in another lecture, but they have compound eyes. Really good at sensing motion, not so great at visual acuity. So here, as a little aside, often you'll find the more predatory an insect is, particularly if they're predatory on the wing, the larger the eyes will be. So dragonflies have got really large eyes because they're flying predators, all right? They're actually catching prey on the wing. Even within flies, we find differences between males and females, because males try and mate with females on the wing, so males have much larger eyes than females that almost touch, so they can get as much perception of area as they can in order to find females to mate with. So if you've got huge compound eyes, likely to be using your eyes in probably predation more than anything else. You don't need big eyes like that if you're just sitting on a plant and eating. Okay, these little things that look like little beads, ocelli, are also visual organs in insects, and we'll talk about them in a further lecture as well. But the number and position of those can help in identifying particular groups of insects, their presence or absence. In this case, we have a little bee here, and she has 3. So it's either combinations of 3, 2, 1, or none. I'll talk about what they do, as I said, in another lecture. Okay, antenna. Antenna gives us a bit of clue too about what an insect might be. We have a number of different ground plans of antenna, unless of course we've gone digital, so there is no antenna at all in that case. That's not the case in insects. I think they all have antenna pretty much of some sort. So this is your basic antenna. Okay, it's got a base called the scape, second segment of pedicel, and this segment here, a flagellum. It's fairly simple. It's whip-like. It's called filiform. It's linear. It's slender. Okay. Then we have another couple of forms here. This one is bead-like or manila form. This one you'll find in termites. And then we start to put a club on the end here, so that's called clavate. We'll have a look at these in the lab. And this one you'll find in certain beetle species. Once we're starting to increase the surface area of the antenna, we'll be starting to think that they're using chemical communication a lot, okay? Particularly in sexual communication between males and females. So we're starting to increase that surface area here. We've got serrate or saw-like, then we've got pectinate or comb-like, and then we've got flammulate or fan-like. So up here, you'll generally find the dimorphisms between males and females, because it's typically females that produce the sex pheromone, and males are the ones that have to find it. So if you maximise the surface area that your receptors can pick it up on, as you could through this, then you can pick up very, very tiny amounts and be the first one to find the female. So you'll generally find differences between males and females when we're getting up here. But all these would suggest some level of chemical communication. Okay, G there. This is like a bent elbow-like. G, geniculate or elbow. We see these in ants in particular. A little club on the end. That's because they're using them as tasters, sort of dropping them down and tasting again with what they might like or otherwise. Now we have another one here which is called feather-like, plumose. This one actually often is involved in sensing movement a bit more, not so much chemical communication, air movement. You might see those on midges and things like that, but not very commonly seen. And the last one here is eye, which is aristate, which is just a bristle. That first segment exists there, and then it's just a bristle off here. And this is generally found in flies, very fast-moving things that fly very fast speed. So you can't have a big old antenna flapping around everywhere in the process. Flies aren't that big for antenna. Okay. Thorax, and this is a reminder really, because I've already said what I probably need to say about the thorax. 3 segments for the thorax. Sometimes it's difficult to pick which segment is which, as it can be in wasps. But having a look here, 3 segments, pro, meso, meta. You can tell them by finding the pairs of legs, and you can tell the meso and the meta by each having a pair of wings associated with it. Now the meso and the metathorax is going to be larger probably than the prothorax in general, because you need more flight muscle in there. So they generally are larger to look at. Legs. Okay, we've got what, 10 minutes to go. That's pretty good. Alright, let's take a look at legs. Now, remember I said that insects are defined not just by 3 pairs of legs, but 3 pairs of jointed legs. So when we put joints, we start to label different sections of the legs. So here we have the coxa, the femur, the tibia, and the tarsi. Okay. In this case, we've got 1, 2, 3, 4, 5 tarsi. Now, tarsal counts can be important when we get to beetles, but we'll do that in another lab. On the end of these tarsi, you generally have claws, and sometimes you have a little pad here, an areolar pad with a number of bristles. So that's so the fly can walk upside down on the ceiling and do all those sorts of things. It's got that pad which is like a suction pad that it can use to do that if they have those pads. Some of them don't, they just have claws. We can see variations, I'll talk about that in a minute. But the tibia and the femur are the two major ones that I want to elaborate a little bit. Okay, so we can change that design in a couple of instances. The obvious one we can see if we've got a very enlarged femur and a swollen femur, if you like, and a very enlarged tibia relative to those, is jumping. That should be fairly obvious to you. This is a grasshopper species where jumping is quite possible. If we were to see a specimen in a museum and it had hairs on its legs, we would suspect that that specimen was aquatic. Okay? And it's a bit like maximising the surface area when you row backwards, but then folding the hairs in to go forward so you're not getting so much friction in your forward motion. So outwards and in. It's a bit like sort of a rowing oar really should be, but you're pulling it out and then back again. Wouldn't have to pull it out if it was based on hairs. Okay, so that is an indication, and we can see here a couple of shots of insects that have these hairs on their legs, like that, which are aquatic. Aha. Jointed appendages. Okay, let's have a just look at the advantage of having a jointed appendage. So if we go back to sort of primitive form, we go back 400 million years ago and find velvet worms. Onychophoran running around on the forest floor amongst the leaf litter. Here's an onychophoran. It's got many, many pairs of legs, if you like, but those legs aren't jointed, okay? You can see they're just a blob on the outside there. So if you were to put an onychophoran in a running race, you could only do about 7 millimetres a second or 0.03 kilometres an hour. Not getting very fast, okay? Let's go to the other extreme. We get up to the fastest of the insects. Tiger beetle, 2.5 metres a second or 9 kilometres an hour. Okay, so you know, put that in a running race against a 100-metre athlete. See how that can't 2.5 metres a second. It would still lose to a human in terms of that by 10, 25 metres. No, 25 metres. And you get a quarter of the way than a human might at that particular point. But if you put it in terms of body length, it's extraordinary. It's 120 body lengths a second, okay, which we couldn't achieve. We're trying to run 120 body lengths a second. So it's 300 times faster than a velvet worm. So the effect of having those jointed appendages has really made a huge difference to the ability to get around and get around fast, chase prey and do all sorts of other things that you might choose or want to do. How do they move around with those jointed appendages? Well, generally they use a tripod approach. So here we have 3 that are in contact with the ground at any one time. So a tripod is maximum stability, okay? Centre of gravity over here, so there's a slight little wriggle each time you move from side to side, but then one set of tripods, they move forward and then this lot become that. So great stability. It's been mimicked, of course, for robot use across multiple terrains because it gives maximal stability having that tripod apparatus. In the last decade or so, when we've got to being really, really fast, high-resolution film, there was a great paper out a little while ago talking about cockroaches running on 2 pairs of legs. So when they get really fast, they don't actually bother to go with the tripod approach, they just go for it, okay? So they can get running for short periods of time on less than 6 pairs of legs, even 2 pairs of legs at one point there, and they're leaping for it. But that process is very good for stability. Okay, let's have a look at the last couple of options if you don't have legs. Well, if you don't have legs, what do? Okay, so here is waves of muscle contraction. This is a blowfly larvae head. These are its hooks. It looks like eyes, doesn't it? But it's actually just sensory apparatus. It looks like something out of a science fiction movie or some sort of comic in some way. But here we have maggots. They don't have any legs, okay? So they have waves of muscle contraction. These waves go from anterior to posterior, and then they have little hooks or tubercles. You can see them here that they go down on, and then they move through again. So things like these, like generally carrion or meat feeders, or in substrates that they're completely surrounded in. That means that you can move through that way quite easily from anterior to posterior. Caterpillars do it the other way around, posterior to anterior ways, okay? So different ways. So here they're bringing the posterior up and then moving the anterior along. And you can see these are the three thoracic pairs of legs, but what they've done is they've developed more legs called prolegs. These aren't real legs, these don't have joints on them, they're not got articulations. They're little fleshy pads, okay, which help them. So you put that fleshy pad down and move forward, etc., like that, Luke. So a posterior to anterior way of muscle contractions. Aha. Wings. Going back to wings in the last few slides here. So let's have a look. So wings, we said, are associated with the meso and metathorax. If we take a look at wings, we get quite a different variety of wings too, externally on insects. So here we look at an aphid, and we can see the forewing is much larger than the hindwing, okay? That can be a pattern we see, or sometimes they can be very similar, like they are in a termite. Okay, a termite sheds its wings after it finds its nesting site, so it doesn't have wings for much of its life. So generally the forewing can be a little larger than the hindwing, as it is with wasps too. Here you can see the forewing are larger than the hindwing. Butterflies, of course, and moths have put a scale on their wing, which gives it a slightly different surface area, and very prominent on those when you look at them. Many of the bugs, the Hemiptera, and certainly the beetles, have changed their forewing, and they've got a hard covering. In the case of many of the bugs, sort of a half covering, which is hemi or half. Beetles, they've got rid of it being a real good flying wing and more cover over the top of their flying wing, okay? So it's a hard cover over the top of that flying wing. When we get down to thrips, as I mentioned briefly in the first lecture, they have they sort of float around in the air, but can get a fair way, but they have fringes all over them. This is the last design that I want to mention. This is flies. Flies have got rid of their hind pair of wings and only have their fore pair of wings. And by doing that, they've managed to achieve very fast flight speeds, and we'll talk about that in the lecture that includes flight. So only one pair of wings. This second pair, when you look at this in the lab this week, is reduced to what's called a haltere or halter. It's like a little chicken drumstick, which acts like a gyroscope for body orientation when they're moving in flight. Last one to look at is the abdomen. So this is the abdomen of the insect. Again, we have 11 segments. What you'll notice on the abdomen is we have plates on the top, tergites, and plates on the bottom, sternites. So these are sclerotized, and in between these you've got that unsclerotized cuticle, endocuticle. So they overlap a bit, so that enables flexibility in the way the abdomen can arch around. And then when you look on the side of every one of these segments, you'll see a little hole on the outside, and this hole is the breathing hole, okay, of insects, the spherical system. They're all spherical holes. So you're able to count the segment, each segment will have a spherical associated with it. And you get a few funky things going on here with the genitalia and fusions of the genitalia and cerci, or little projections hanging out the back for sensing movement and other things. So that's the basic ground plan. Okay, I'll finish that one up for you by putting it to stop capture. Now, with the labs, you should have